Hello and welcome to our second lecture of the course. This is all about brain and behavior. So in this chapter what we're really focusing on is the nervous system, the brain, the spinal cord, and looking at the biological bases of behavior. And this is really the area of psychology that I focus on, looking at the biological mechanisms of learning, memory, and why it is we do what we do in terms of brain activity. So the nervous system is made up of the central and the peripheral nervous system. So these are the two major divisions of the nervous system. The central nervous system is made up of the brain and the spinal cord. If you were to look at a human right here, the brain and the spinal cord, these lie along the midline. So that's very central to the body, right? That's an easy way to remember what lies in the center. The peripheral nervous system is everything else outside of the brain and the spinal cord. So this is composed of the somatic nervous system. The somatic nervous system is important for our voluntary movement, muscle activation, so information that gets sent out to the muscles to activate them, and information that comes in through the senses in terms of like touch go into the spinal cord. That's examples of the somatic nervous system. The other division of the peripheral nervous system is the autonomic nervous system. Now, as the name suggests, the autonomic nervous system takes care of things that are automatic, involuntary, things that we don't really want to have conscious, voluntary control over, like whether or not your stomach is secreting enough gastric juices, enough enzymes, if your small intestines are pushing food through. You don't really want to have all that conscious activity going on because Lord knows you would forget and you die. So the autonomic nervous system is made up of two parts: the sympathetic nervous system, which is our fight or flight system, and our parasympathetic nervous system, which helps to bring us back down to resting state. Now the individual nerve cell in the brain is known as a neuron, and a nerve is a bundle of numerous axons, and axons are large projection or long projection neurites that arise from a neuronal cell body. And you have two different types. You have cranial nerves and you have spinal nerves. So here are the basic divisions. Again, the central nervous system is made up of the brain and the spinal cord, and this lies along the midline. So this is in the center of the body. Think about that. The peripheral nervous system is the somatic nervous system and the autonomic nervous system. Then the autonomic nervous system is made up of the sympathetic and the parasympathetic. And here are just examples and descriptions of what each of these goes to. This will be very important for your test, particularly in looking and understanding what divisions are in what area of the nervous system. So neurons, these are your brain cells. You can tell that they look a little different than the way normal cells are depicted in the body. And there's around 85 billion of them within our nervous system alone, with each neuron making several thousands of connections, or a range between hundreds to thousands of connections. So for every neuron, there's approximately 10 glial cells, and these are cells that are playing numerous supporting roles in and around the neurons. So potassium spatial buffering, providing additional nutrients, controlling neurotransmitter activity, etc. So the basic parts of a neuron that are going to be important for you to understand are the dendrites, which are the receiving end of a neuron. So if you think about like the entrance to a home, this would be the door. The dendrites are the part where information will be able to come in. The soma, which is the cell body. This contains the DNA and all the organelles that you would normally find in a regular cell. The axon, which is a thin fiber that leaves the soma. It's a projection neurite. So information is going to be sent down the axon to the axon terminals, which, as the name suggests, is the endpoint of the axon. These are bulb-shaped endings where chemicals can cross a synapse. And a synapse is just a space between the terminal of one axon and most often the dendrite of another neuron. So it's a very small gap where chemicals called neurotransmitters will actually cross here and bind to receptors. So right here. Right here, this small connection is what I drew up here. Okay, So whereas when I teach physiological psychology, I spend about an entire lecture, or 2 lectures in the case of brain and sensory processes, covering the nerve impulse. Well, because it's Psych 100, I'm going to go through it in about 1 to 2 slides. So when a neuron is in resting state, and what this simply means is that the neuron is not actively firing, it's more negative inside than on the outside. So the inside of the cell is at a resting membrane potential of negative 70 millivolts. These are due to ions and proteins that are inhabiting the outside and inside of the cell. Negative 55 millivolts is known as the activation threshold. Now, the way that you have a change from negative 70 millivolts to negative 55 millivolts is through depolarization. So your cell will be able to bring in ions such as sodium, which has a 1+ charge. And the more sodium that flows into the cell, the more positive or less negative the membrane potential gets. Now, once negative 55 is achieved, a neuron will fire an action potential. This is an all or none response, and an action potential is known as the nerve impulse, and it is the signal that is going to be carried down the axon. So this is a depolarization that occurs from negative 70 millivolts, again, due to the movement of ions such as sodium, or these charged atomic species. So sodium rushes into the neuron, further depolarizing the cell, simply making the cell more positive. When the cell is at about positive 40 millivolts, the sodium channels will shut because they're voltage gated, and no more sodium will actually be able to come into the cell. Potassium channels will open at that point, allowing for potassium to leave. This leads to a negative after potential of roughly negative 80 millivolts. So this is a drawing that I made when I taught Psych 100 a few years ago. So here we have ions such as sodium and potassium. This is the resting membrane potential at negative 70 millivolts. Something happens. A neurotransmitter binds to a receptor, let's say, and brings in sodium ions. This is a depolarization because you are becoming less negative or more positive in terms of membrane potential. At negative 55 millivolts, this is the activation threshold. This is when your voltage-gated sodium channels will open, causing a massive depolarization. Now, at about positive 40 millivolts, your sodium channels are going to close. At this point, and roughly down here, your potassium channels are going to open, and the cells are more positive. At this point, you're above 0, so let's say 0 is right here. Potassium is going to want to leave the cell. So as potassium leaves, it carries a 1+ charge. The more positive ions that flow out, this is going to cause a hyperpolarization, meaning you're going to become more negative or less positive. Now, these channels are going to open and stay open until about the negative after potential, which is roughly around negative 80 millivolts. And at this point, the cell, through the use of the sodium potassium pump, will help to restore the balance of ions within the cell. So you'll get back to the resting membrane potential. I recommend reading about this in your book, as well as watching this particular explanation a few times to be able to really grasp this concept. Again, I normally spend whole lectures explaining the resting membrane potential, what's actually going on in the action potential, but because it's Psych 100, I'm not going to go too heavy into detail. But for this class, you should understand the basic idea of why the action potential exists. So every cell, in terms of your neurons that have a resting membrane potential, are going to fire once a certain level is reached, and that's what's known as the activation threshold. Now, in your book, your threshold is set at negative 65, which is odd because I've rarely seen that used. However, every cell is a bit different in terms of what is activation threshold and what is not. But what causes there to be a massive depolarization is sodium channels opening. So you need to understand that sodium channels open, causing for sodium to rush in and a depolarization to occur. At about positive 40 millivolts and positive, your potassium channels are going to open and potassium will start to leave the cell. Sodium channels close at positive 40 millivolts. And down here at the negative after potential, your potassium channels are going to close. What brings this back to the resting membrane potential is the sodium-potassium pump. Now the sodium-potassium pump helps to restore this balance of ions by taking out 3 sodium ions and bringing in 2 potassium ions. It's a pump because it requires energy in the form of ATP to take ions against their concentration gradients. Myelin is an electrically insulating material that covers a majority of the neurons within the nervous system, and it covers the axons. So if we draw a neuron like this, here are some dendrites, and then we draw an axon, and then here's a terminal. Myelin is an insulating material that will cover some neurons within the nervous system. This is an electrically insulating material, and this actually helps for action potentials to travel quicker down the axon. Now there are small gaps within the myelin sheath, and these are known as nodes of Ranvier. And what's interesting about the nodes of Ranvier is that they contain a large amount of voltage-gated sodium channels, allowing for the nerve impulse to actually be regenerated across and at each node. This allows for quicker transmission. So these nodes help the nerve impulse to move quicker, and the action potential leaps from node to node. Hence the name saltatory conduction. Multiple sclerosis is an example of a disease where the immune system actually attacks and destroys the myelin sheath, causing disruptions in not only vision but also movement. And if you think about it, because myelin is allowing for faster nerve impulse conduction, if you impair that in a normally myelinated axon, that's going to greatly impair whatever information is normally traveling at a quick pace, such as visual information in the optic nerve. So one important distinction to note in the nervous system is the difference between gray and white matter. Gray matter contains somas and dendrites. White matter contains myelinated axons. Now, why is it white matter? Myelin is a fatty covering, and so if you think about like the fat that lies in steaks, it's a creamy color, it's whitish. That's hence why it's white matter. It's the component of those myelinated axons. So here's a myelinated axon. Here's your myelin sheath that is wrapped around the axon. Here's a node of Ranvier right here, which is an unmyelinated portion of an axon. Your current will flow underneath the myelin to be regenerated at each node of Ranvier. This is in comparison to an unmyelinated axon, which do exist in the nervous system, where this actually has to occur, this charge distribution has to occur along every single stretch of the axon, compared to only occurring at each node. That's why saltatory conduction, as seen in this first example, is a quicker method of transportation for the action potential. The communication that occurs between two neurons is chemical, for the most part. So in the synaptic cleft, you have an exchange of small chemicals known as neurotransmitters. The synapse, which is right here, is this small space that lies between a presynaptic and a postsynaptic neuron. Presynaptic just simply means that it comes before the synapse. So cell A is our presynaptic neuron. And so here to the cell body, dendrites. And then our postsynaptic cell is the receiving cell. This is the cell that lies after or post to the synapse. Neurotransmitters are released into the synaptic cleft when an action potential arrives at this presynaptic terminal. So the action potential arises from the axon hillock and will travel down the axon, and when it gets to the terminal, this will lead to the release of neurotransmitters in the process known as exocytosis. Neurotransmitters are then able to attach to receptors that lie on the postsynaptic neuron. So here I'm going to draw cell B. Cell B has receptors that will be able to recognize particular neurotransmitters. And when neurotransmitters, which are found in the cleft after being released from cell A, bind to receptors, this will lead to channels opening, allowing for ions such as sodium to flow in. Other receptors allow for things like potassium and chloride ions to flow in. So neurotransmitters change the likelihood of an action potential. Meaning, again, if you remember back to our action potential that we covered a few minutes ago, you need to reach -55 in order for an action potential to occur. If sodium is flowing in, this is going to cause the membrane potential to increase. If you bring in something like chloride ions carry a negative charge. This will bring the resting membrane potential down, or more negative, taking you further away from the point where you need to be in order to fire an action potential. So some neurotransmitters are excitatory, such as glutamate. Some are inhibitory, such as GABA, and others play modulatory roles in either excitatory or inhibitory fashions depending on what type of receptor these neurotransmitters bind to, such as dopamine and serotonin. So here's another drawing that I have done for Psych 100. These are dendrites. Incoming information will go into the dendrites. This information will travel down to the soma, which is the cell body, which contains our organelles. This segment right here is known as the axon hillock. This is the part of the neuron where if you reach -55 millivolts, an action potential will fire and travel down the axon. Here's our axon that is composed of myelin sheath and nodes of Ranvier. When the action potential gets down to the terminal, right here, it will release neurotransmitters into the synaptic cleft, which is the space between the pre-synaptic axon or cell and the postsynaptic cell. So here's another image from your book. This connection between an axon terminal and a dendrite of a receiving neuron. You have the axon terminal, synaptic vesicles which contain your neurotransmitters. Neurotransmitters are then released in the process of exocytosis. They can bind to receptors that lie on the postsynaptic membrane, allowing for ions to flow through, affecting the likelihood of an action potential in that next cell. So releasing the neurotransmitter in the process of exocytosis is composed of a few steps. So the action potential, like we've said, reaches the axon terminal. These vesicles are released from their protein anchors. The vesicles fuse with the axonal membrane to release their neurotransmitters. Then the vesicle material is then recycled and is refilled with neurotransmitters. And neurotransmitters, small neurotransmitters anyways, are actually synthesized right at the point of release. Now, certain drugs are able to mimic, duplicate, or block neurotransmitters from binding. Blocking neurotransmitters would be an example of an antagonist. Mimicking would be an example of an agonist. Cocaine, the drug cocaine, has a very similar chemical structure to dopamine, and DA is the abbreviation for dopamine. The use of cocaine can increase dopamine in the reward system by essentially causing dopamine to flood out of the neuron, allowing for more and more dopamine to bind to more and more dopamine receptors, causing you to feel really, really good. So drug addiction can occur, and basically what drug addiction does is it hijacks the reward system that is normally in place. So if you do something enjoyable, like score a goal in soccer, or save a goal in soccer for all my goalies out there, if you do something enjoyable, like eat a burger, have sex, go to an amusement park, dopamine is released to make you feel good. Now, obviously, drugs make you feel a hell of a lot more good than just eating a hamburger, right? Do not do drugs. They are bad. But why people get addicted is because this essentially hijacks the natural reward system of your body, and your natural rewards can't compete with the reward system that dopamine released through drugs can provide. Curare is an acetylcholine antagonist, and an antagonist will block the neurotransmitter from binding. And we'll get into a lot more detail of this in Chapter 5 when we talk about drugs. But an antagonist blocks a neurotransmitter from being able to bind to a particular receptor. So acetylcholine can no longer act on receptor sites in muscles, and this will lead to paralysis. Neuropeptides are small proteins, which are chemicals that can help to regulate the activity of several neurons. And so a few examples include enkephalins, which help in relieving pain and stress, and endorphins that play a role in reducing pain. So a few examples may be the placebo effect, which we talked about in chapter 1, and even the sensation of runner's high can be influenced through the role of endorphins being released through the body. Neural networks are interlinked collections of neurons that help to process information in the brain. A simple neural network example is a reflex arc. So this occurs when a stimulus provokes an automatic response. So no brain activity is required for a reflex arc to occur. However, the brain is responsible for the processing of this information. So if you were to touch a hot stove, your hand is going to automatically jump back. What makes you yell explicit words or expletive words is your brain processing that painful information. Now, why is this a good thing in terms of no brain activity being required for these arcs to occur? Well, it frees up other neural networks. Our exclamations are controlled by the brain in our perception of that stimulus. So in the case of the example of touching something hot, it's our perception of pain. So here's an example of a simple reflex arc, touching something hot. If you touch something hot, a sensory neuron will carry that information up to the spinal cord through our dorsal root ganglia, making a connection to an interneuron. Here, making a connection to a motor neuron, the motor neuron making a connection to the muscle, causing our arm to flex up and be removed from that painful stimulus. So the sensory neuron again brings that painful stimulus. The motor neuron sends the command to the muscles to yank our arm away, and an interneuron is a neuron linking other neurons that are found within a given structure. So an interneuron has all of its processes within a given structure. In this case, it's within the spinal cord. Now, information can then be integrated into the nervous system in a few different ways. So we've talked about neural networks already, and your neurons are all making connections to thousands of other neurons. So you don't just have often one-to-one communication occurring in the brain. You have a cell receiving information from hundreds or thousands of different cells, and it has to integrate that information in a way where, again, if you reach negative 55, you're going to fire. If you don't, you don't, because it's an all or none response. So a few different ways that information can be integrated is through temporal summation and spatial summation. So in temporal summation, action potentials leading to the release of signals at a single location on a receiving neuron occurring many times influences the receiving neuron. So here is a presynaptic cell sending action potentials in close time. So one right after the other onto a receiving cell. So in this way, it summates over time, or it sums over time. So if just A occurred, so action potential A, if just action potential A occurred, it didn't quite get to negative 55, so it might have actually just gone back down to the resting state. However, if A is followed by B, which is then followed by C in close succession in time, hence temporal, these can add together to get to negative 55, causing an action potential to then occur. Spatial summation is when action potentials from different spaces or different locations on a receiving neuron influence the receiving neuron. So again, if A had just occurred, so if this connection was the only connection, let's say, you might have had a small depolarization, but then it would have gone back down to resting state. However, if A and B and C cells all connect to, let's call this neuron D, at the same time, but across different spaces and locations, these can then add together to reach negative 55 millivolts, causing the cell to fire. Neuroplasticity is the ability of our brain to change in response to experience. So the brain is plastic. You're not just stuck with the same brain and the same neural connections. This was known as Hebb's rule, which is the repeated activation of synapses between two neurons strengthens the connection between them. And so we'll talk more about this when we discuss learning and memory. But our brain is very plastic in terms of the fact that it adapts to our environments, what we're bringing in, what we're learning, what we're remembering, etc. So your synapses are going through constant changes. So you can see this in rats raised in enriched environments versus rats that are raised alone with no enriched environments. That's often why in animal research we definitely make sure to house our animals in cages of other animals as well, so they're not alone. This can change the way of the actual structure of the neuron itself to increase the surface area, having increased dendritic spines or increased dendritic complexity. Neurogenesis is the production of new brain cells. So these cells are most likely involved in learning and memory and our ability to adapt to changing circumstances. So when you're born with all the neurons that you're going to have. You don't just have, like when you get a scrape, you have new cells that are able to be made to be able to form a scab and be able to heal itself, right? You don't have that in the nervous system. You don't have cells that go through mitosis. You have all the cells you're going to have, but there are areas in the brain, including the hippocampus and in the olfactory system, where you do have regeneration of cells that are occurring. So an important part of psychology is being able to understand and look at different ways that we can measure brain activity. So here I've made a chart where we have the imaging technology on the left, the procedure that's occurring with this technology in the middle, and then what questions we can answer using that technology on the right. So this is just a brief summary slide, and we'll go through examples of each, but computerized tomography or CAT scans uses X-rays taken from different angles and essentially collected to form an image. Now, the questions we can answer are what regions of the brain did a stroke impact, per se. CAT scans help to reveal brain structure. Magnetic resonance imaging uses magnetic fields to form 3D models of a particular brain or body. And the questions that we can ask are similar to a CAT scan in what does this person's brain look like? Are there any structural abnormalities? Now, magnetic resonance imaging provides a much clearer picture than a CAT scan in terms of looking at the brain, and you don't have X-rays to deal with. EEGs, or electroencephalograms, use electrodes to help track electrical activity. So the questions that we can ask and what we often use in sleep research is, what is a person's state of arousal? What stage of sleep is somebody in, based on the way that the activity in the brain is going and doing. Positron emission tomography and functional magnetic resonance imaging, these are functional tasks. So positron emission tomography is where we take radioactively tagged glucose or oxygen, and this helps us to track chemical activity in the brain. This is due to the idea that, and to the reality, that areas of your brain that are more active, those cells are requiring more glucose and more oxygen for that increased activity. So what questions we can ask and answer are, what parts of the brain are active during a particular task? And these go for both of these technologies. Now, compared to positron emission tomography, fMRI uses magnetic fields as well to help track the flow of oxygen. So if you were to be put into an fMRI, someone shows you a very scary image and you react, we're most likely going to see activity in the amygdala, which is a brain area that's important for not only stress, but also processing of fearful or stressful images. So an MRI helps to show structure of the brain, very clear structure, and then a PET scan helps to show function. So seeing, in a seeing task, we have more activity in the occipital lobe. In a hearing task, we have more activity in the temporal lobe. Thinking, we have activity in a majority of the brain, in a lot of areas of the brain. And in speaking, we have activity here within the brain as well. EEGs use electrodes that help to track electrical activity in the brain. fMRI shows different areas of the brain that are lit up when individuals are in a certain task. So one interesting thing to note is that in the case of lying and telling the truth, your brain actually shows different activity. So it's something more so in the ethical realm of should this be used as a lie detection method? Because if you think about it, the more you lie, you have to keep track of more things in your memory, right, in order to maintain the lie. So compared to telling the truth, you actually have different activity levels in the brain when you're lying versus when you're telling the truth. So a few additional technologies. Electrical stimulation, or ESB, of the brain. This delivers electrical currents through an electrode into a given area. So the questions that we can answer are, what happens when region X in the brain is stimulated? Ablation is a surgical removal of parts of the surface of the brain or brain areas. So the questions that we can answer are, what happens when this part of the brain is removed? Obviously, we're not doing this to human patients like volunteers. We can often do these experiments with rodents or lower species. Deep lesioning are the surgical removal of parts of the brain that are deeper than just the surface. So we can say what happens when some deep structure of the brain is damaged. And again, we're not doing this with human participants. In the case of ablation or deep lesioning, oftentimes we can use case studies. So if you remember back to our first lecture, we could use case studies to be able to look at individuals who happen to have areas that have been either removed due to necessity or due to injury. And microelectrode recording is measuring electrical activity of single neurons. So oftentimes this is used in vitro research when we plate neurons into a cell dish, and we can look at what individual neurons are like and what their activity is like. Now, human brains aren't the biggest. We don't have the largest brains in the animal kingdom, and we don't have the best ratio of brain to body weight. Now, what actually makes humans very unique in terms of our reasoning and intellect is our cerebral cortex, the outer layer of the brain. Cortex is actually Latin for bark. So if you think about a tree bark. Now, the cerebral cortex contains about 70% of the neurons that lie within the central nervous system, and it's responsible for many of the things that, again, make us uniquely human. Our ability to use language, live in complex social groups, and to acquire and use complex skills. If you were to look at a brain, we have a very wrinkly cortex, with many sulci, which are known as ridges, and gyri, which are known as grooves. So here is a gyri, or a gyrus, singular. So let's say this is a gyrus. Gyrus here. So these grooves. And then a sulcus will be these lines here, these deep lines, these folds and ridges. The cerebral cortex is composed of two sides, the cerebral hemispheres, left and right. If you were to look at a brain, from the dorsal side, which just means from the top, you have a left and a right side. These two hemispheres are connected by a thick band of fibers known as the corpus callosum. This allows for communication to occur between the two hemispheres. The right hemisphere mainly controls the left side of the body, and vice versa. The left controls the right side of the body. The cerebral cortex is further divided into smaller areas, and these are known as the lobes. And I am sure all of you have probably heard of the lobes of the cerebral cortex. The frontal lobe, the temporal lobe, the parietal lobe, and the occipital lobe, and it's important before you leave this class to have an understanding of these lobes. And we will discuss the different lobes in a few minutes. Now, our cortex displays lateralization. And what this means is that each hemisphere has specializations for particular abilities. Now, this is not exclusive. It's not saying that certain hemispheres contain only the ability to do certain things. But what we often see is language is lateralized to the left hemisphere, and emotions more so lateralized to the right hemisphere. This does not mean that, again, language is only in the left and emotions only in the right. These are just examples of a few ways that our brain and cortex has been lateralized. So here are just a few brief examples of the brain where the locations of the lobes are. Here again, this is a sulcus. This is the central sulcus. And then this whole thing right here is a gyrus. right here. This example is showing how the left side of the brain controls the right side of the body. And then here is the corpus callosum, which is this thick band of white matter that is connecting the two sides of the brain. This is a mid-sagittal cut right here, and our cortex is this area here. Now, how can we test hemispheric lateralization? Well, there are actually not natural people, but there are actually test subjects that we can use this, and these are split brain patients. Now, in order to control severe epilepsy, individuals would often have their corpus callosum severed, and some still do today. Now, what happens is, again, going to this image right here, the corpus callosum is connecting both hemispheres. In split brain patients, these patients undergo surgery which sever the corpus callosum. So input from the left field of view is processed by the right hemisphere, and we'll talk about this in chapter 4 when we go through sensation and perception. And information on the right side of our visual world is processed on the left. Now, this is when it gets really interesting and really fun. So in split brain patients, you have one patient right here that has undergone severing of the corpus callosum. A word is flashed briefly to the right field of view, and the patient is going to be asked to say what he saw. So on the right side, this is going to be processed on the left. Because the left hemisphere is dominant for verbal processing and language, the patient's answer matches the word. So he says face. Now, a word is flashed on the left field of view, and the patient is asked to say what he saw, and he says nothing. This is an example of lateralization where language isn't dominant in the right hemisphere. So the right hemisphere can't share that information with the left hemisphere at this point. So the patient is unable to say what he saw. But, interestingly enough, he can draw it. So experiments with these split brain patients have helped to illuminate the lateralized nature of brain functioning. Now, we can't go into the lab and just sever corpus callosums and do all these experiments, right? But for individuals that need this in order to help to control epilepsy, we can use them as essentially as sort of case studies in looking at how split brains are leading to lateralization, or not necessarily leading to lateralization, but how they exemplify lateralization of the cortex. So, are you left-brained or right-brained? Well, the answer is we aren't exclusively left or right-brained. I know that you've probably taken a quiz sometime in your lifetime that has told you if you're more right-brained or more left-brained. The idea here is that both hemispheres of your brain work together to help provide a collective experience. You're both analytical and emotional, you're creative, and you can write and talk. The idea of lateralization is that the left hemisphere is more, tuned in with analysis of information, language production areas, language comprehension areas, writing, among others, is found more so and lateralized into the left, whereas the right processes information more holistically, as well as recognizing and expressing emotions and processing of nonverbal aspects of communication. So here I've provided a link that will take you to a website where you can actually test whether you're more left-brained or right-brained, but our big consensus here is that you aren't exclusively left or right-brained. Your whole brain is working, right? So remember that we use both hemispheres, but some tasks may make more use of one hemisphere versus another. So if you were to be asked what this image is, your left hemisphere would say a bunch of D's, whereas the right hemisphere, which processes overall patterns, would process this as the letter L. If you were to be asked about this expression, a stitch in time saves 9, your left hemisphere, which is more detail-oriented, would say it's about sewing, whereas the overall pattern in the right hemisphere is a small effort now saves time later. Right hemisphere processing holistically can say that this is an eye, where your left hemisphere says it's made up of a bunch of dots and blobs. So your brain works together to put together these images and be able to process this sensory information. So are the brains of men and women different? Although the implications remain to be understood, there are some physical differences between male and female brains, even differences in the ways that connections are arranged. So both men and women utilize the left hemisphere for language tasks. However, the left and right hemispheres of women's brains were more activated, or were activated more in half of women tested. So this could have implications in behavioral differences between the sexes, and the ability of speech to be regained differently between the sexes. Okay, so here are the lobes of the cerebral cortex. Your frontal lobe, based on how it sounds, is in the front. So if I draw a brain like this, central sulcus, the frontal lobe is in the front, and this is known as anterior in directional terms. The frontal lobe is associated with higher order cognition. Due to the presence of the primary motor cortex that lies in the frontal lobe, known as M1, this helps to control control movement. This also contains what are known as mirror neurons. In the left frontal lobe is Broca's area, and in Broca's area is important for production of speech. If you were to damage Broca's area, this leads to aphasia, the inability of you to be able to speak. Another big aspect of the frontal lobe is what's known as the prefrontal cortex, which is related to very complex behaviors, personality, reasoning, our ability to plan. Now, let me change my color to blue. The parietal lobe is dorsal, meaning that it's on the top. It's posterior to the frontal lobe, and so we're going to draw the posterior or the parietal lobe here. It's anterior to the occipital lobe, which lies right here. Anterior meaning in front of. The parietal lobe processes somatic sensations, so touch, temperature, and pressure. And it also is the location of our primary somatosensory cortex, which is known as S1, which will process that incoming touch, temperature, and pressure somatic sensory information. Now these areas, or these illustrations, are what are known as homunculi. So your primary motor and your primary somatosensory cortex have different densities of neurons that are receiving information about particular areas of the body. If you think about your motor cortex and you think about your somatosensory cortex, you can think about certain areas of the body that are more responsive in terms of sensation and more adapted to particular fine movements. Luckily, we don't look like this, but this is what you would look like if the density of information that is allocated to our cortices were to be distributed to us as human beings in terms of body proportions. So note that we have huge hands in both motor as well as sensory homunculi. That's because in the motor system, we're able to do fine-tuned movements, be able to do quick movements with our fingers and grasp objects. So we have very large amounts of our cortex devoted to information from our hands, and to be able to touch and feel in the case of the somatosensory cortex. We have very small feet. Roughly speaking, if you're asked to judge distances between two stimuli, you're often better able to do it with your hands versus your feet. Your trunk is often very small in these homunculi drawings due to the fact that how often do you go around the world feeling things with your chest or your stomach? And then your lips for fine movement of speech as well as for sensing information coming in are often pronounced in the homunculi as well. Okay, so let me draw a brain, so cerebellum, spinal cord. Your temporal lobe lies to the side of the brain, near your temples. This contains your primary auditory cortex, known as A1. This processes auditory information. The left temporal lobe contains Wernicke's area, which is important for speech comprehension. So difficulty in understanding the meaning of words will occur if you were to have damage to the left temporal lobe, particularly in Wernicke's area. The occipital lobe, and I'll change colors to, let's do yellow. The occipital lobe is posterior to the frontal lobe, so it's here. It's posterior to not only the frontal lobe, but also the parietal lobe. This contains our primary visual cortex, also known as area V1. Visual agnosia can result in damage to the occipital lobe and difficulty in naming visual objects. Face blindness, damage to the fusiform face area, which is actually a further target of information from our occipital lobe. And blind sight can occur in individuals with damage to the primary visual cortex. Blind sight is seeing without conscious awareness. Now in this video, you have a patient who is blind, and the reason that he is blind is because he has damage to the occipital lobe. So information is still able to come in through the eyes. However, the part of the brain that is processing that information is gone, right? So even though you bring in this visual input, you actually have nothing to make sense of it, therefore you're blind. So this patient was asked to walk down this hallway, and researchers placed objects in the hallway. He was not told that there were objects in the hallway, but he naturally avoided those objects. Blind sight is the essentially seeing without conscious experience and awareness, and this is due to several other pathways of visual information, and we'll briefly talk about that in chapter 4. Let me pull up here. So now we're going to talk more about some internal cerebral structures. So the hindbrain. The hindbrain consists of the medulla, the cerebellum, and the pons. The brainstem consists mainly of the medulla and the cerebellum, as well as the pons and a few other midbrain structures as well, depending on who you're listening to in terms of defining what the brainstem is. Some people include all the way up to the diencephalon. The medulla controls numerous vital life functions. So if you were to have damage to the medulla, you're often dead. The medulla contains our ability to autonomically allow for breathing, heart rate, respiration, etc. If you have damage to that, you're often not going to survive. The cerebellum helps to regulate posture, muscle coordination, aim, balance, and timing, as well as allowing for memories for skills and habits. The reticular formation flows through the hindbrain. This lies inside the medulla and the brainstem, and it helps to influence attention and is very important in arousal, so keeping us vigilant, alert, and awake. So here, again, is a depiction of our mid-sagittal cut, our medulla here, the cerebellum. Let me change the pen color to something a little bit more visible. Cerebellum. This is the pons, and the reticular formation, which runs up through the hindbrain or the brainstem. Now, if you've ever seen the Powerpuff Girls, I'm sure you remember Miss Cerebellum, the assistant to the mayor. This looks like her hair, right? Very poofy, very large. So cerebellum. Moving a little bit into the forebrain, you have the thalamus and the hypothalamus. Collectively, these are referred to as the diencephalon. The thalamus acts as a sensory relay station for sensory information going up to the cortex. All of your senses go through the thalamus before going to the cortex, except for olfaction, and we'll get into that in a little bit more detail in chapter 4. The hypothalamus is a master control center for many basic motivational behaviors, including hunger, thirst, and other visceral body functions. Here we think of the 4 F's in terms of hypothalamic functioning: feeding, fighting, fleeing, and fornicating, or some other F word if you would like to think about it. So these are collectively referred to as the diencephalon. So the forebrain can actually be divided into 2 different areas: the telencephalon here, and the diencephalon, which is composed of the thalamus and the hypothalamus. The telencephalon includes our cerebral cortex, as well as the olfactory bulb, as well as the cingulate gyrus and the basal telencephalon down here. Still remaining in the subcortex, the limbic system consists of the amygdala, the hypothalamus, and parts of the thalamus, the hippocampus, and other brain structures. Now, the limbic system plays a major role in producing emotion as well as motivated behavior. The amygdala is strongly related to fear and the memory of fearful experiences. Now, things with a lot, and this should say things with lots of emotional connection. The amygdala helps to place an emotional tag onto certain sensory experiences. The hippocampus, which is Greek for seahorse, is critical for forming memories. And we talked a little bit about patient HM last time, where he had his medial temporal lobes removed, removing his hippocampi and causing him to be unable to form new memories. The hippocampus is housed within the temporal lobes, and it is very important for spatial memory, as seen in animal studies that have Alzheimer's, and Alzheimer's impacts the hippocampus. And we see that in animals, damage to the hippocampus will often result in huge deficits to spatial memory. The limbic system is involved in not only fearful activities, but also pleasurable, aversive events, and drug cravings. So the limbic system is composed of a lot of different brain structures. So the hypothalamus, mammillary bodies, the thalamus, the fornix, the pineal gland, the hippocampus, parahippocampal gyri, the cingulate gyrus. So depending on what source you're reading, some individuals will keep some structures in the limbic system, whereas others will classify other brain structures to be out of the limbic system. I'm not going to ask you in this class to list structures of the limbic system, but you should be familiar with some of the major ones, particularly the amygdala and the hippocampus. The olfactory bulb is often also involved in the limbic system. So here, the hypothalamus, amygdala, hippocampus, cingulate cortex, and then basal ganglia, amygdala, and thalamus, showing some more subcortical structures. The endocrine system is made up of glands that secrete chemicals directly into the bloodstream. These chemicals are known as hormones, and they're carried throughout the body that have the ability to affect internal activities and visible behavior. So hormones are related to neurotransmitters in the way that these are peptide neurotransmitters that are able to actually not go through directed synaptic transmission, but be released over large sites or farther sites. So the hypothalamus and the pituitary gland act in synchrony to be able to lead to hormonal processing and release through different glands throughout the body: the thyroid gland, parathyroids, the testes, pancreas, adrenal glands, ovaries, allowing for hormones to be released, even at given moments in time. So the pituitary gland is often known as the master gland due to its influence over other endocrine glands. The pituitary gland is directed by the hypothalamus that lies superior to it, which just simply means that it lies above it. One important role is the control of growth, such as growth hormone release. Oxytocin is a great example of a neurohormone. This is known as the cuddle hormone. I actually have a tattoo of oxytocin on my side. But also oxytocin plays a very large role in pair bond formation, as well as that sense of well-being with friends and family and connections that we feel with other people. The pineal gland releases melatonin, in response to variations in light in our circadian rhythms, and we'll talk about this more in Chapter 5 when we talk about sleep. The levels of melatonin rise at dusk, they continue to rise, they peak at around between midnight and 3 a.m. and fall as the morning approaches. So these are going to be in response to variations in outside light. So the pituitary gland is the master gland, and this is going to have impacts on the release of hormones and the effects at different glands within the body. Interesting fact, the pineal gland, which controls and allows for melatonin to be released, is the only brain structure that exists in one. So you'll have a left half and a right half of everything. You have frontal lobe, frontal lobe. Pineal gland, you only have one inside the brain. So here's the pituitary gland and the hypothalamus that lies right above it. The hypothalamus will then send information down to the pituitary. The pituitary can then send information to all different areas of the body, further away from the direct site of release. So the thyroid gland helps to regulate metabolism and can have effects on personality. The adrenal glands release epinephrine and norepinephrine, and these are also known as adrenaline and noradrenaline. The adrenal glands are located on the top of the kidneys. This is the source of epinephrine and norepinephrine, particularly in the periphery. So here are the kidneys, and then in a rat, you have a tiny little adrenal gland that lie right on top. Now, the adrenal glands produce hormones called corticoids and glucocorticoids, and these are examples of stress hormones, such as cortisol, that's released in times, particularly in our fight or flight system. So here we have the kidney and the adrenal glands, which are then able to release glucocorticoids such as cortisol, epinephrine, and norepinephrine. So the last slide here, looking at the functions of different hormones that lie within the body. So endorphins are endogenous morphines that modify our natural response to pain that can act as neuromodulators to brain activity. Melatonin, which is released by the pineal gland in response to daily cycles or circadian rhythms of light and dark. Epinephrine is an adrenal hormone that tends to arouse the body and is often associated with fear. Norepinephrine is a neurotransmitter and an adrenal hormone that tends to arouse the body and is associated with not only fear and anger, but also stress. And then thyroid hormones, which help to regulate metabolism rate. So that's it for Chapter 2. In Chapter 4 and 5, we're going to be going through a lot more in-depth biological psychology, particularly as a result of looking at the processing of sensation in all of your sensory systems, as well as looking at the perceptual effect of top-down processing, and then going through, in Chapter 5, drugs and sleep, looking at states of altered consciousness. So I hope that you've enjoyed our brief intro to biological psychology, and I'll see you in the next few chapters.